Greetings to all. I'll be presenting the research project entitled Supersaturable Bio-Self-Nanoemulsifying Drug Delivery System for Improving Oral Bioavailability of Dasatinib. Dasatinib is a BCS class 2 drug having poor aqueous solubility and poor oral bioavailability when administered in the form of conventional oral dosage forms. So, we use SNITs as a formulation strategy to overcome the drawbacks associated with dasatinib delivery. Supersaturable biosnits consist of the drug dissolved in a combination of synthetic oil and a bioactive oil with aid of a surfactant and co-surfactant. Along with this, a precipitation inhibitor is also incorporated to improve the drug loading without causing precipitation. This anhydrous preconcentrates when they come in contact with the aqueous GI environment, and due to the peristaltic movement, they'll form emulsion globules inside the body. These emulsion globules will be subjected to digestion. The digestion products like monoglycerides, triglycerides, and fatty acids will form micellar and vesicular structure with the drug. These colloidal structures will act like a reservoir and they will enhance the solubility and permeability of drug. They also promote lymphatic transport. The main challenge over here is supersaturation. Since our drug has pure solubility in intestinal pH, it will be present in a supersaturated state and hence highly prone to precipitation. In order to prevent this precipitation, we are incorporating a precipitation inhibitor. These are the major objectives of the study. To start with the selection of excipients, we performed solubility studies in various excipients. Among the synthetic oils which were screened, dasatinib showed maximum solubility in oleic acid. And among the natural oils which were screened, dasatinib showed maximum solubility in chamomile oil. Hence, oleic acid and chamomile oil were selected as the oil components of our formulation system. Further, surfactants and co-surfactants were screened based upon their emulsification ability. For this purpose, emulsions were prepared with various surfactants, and the prepared emulsions were characterized for size, PDA, and transmittance. Based on these results, Chromophore RH40 was selected as the surfactant. A similar protocol was followed for screening of co-surfactants also, and transcutol HP was selected as the co-surfactant. After selection of excipients, we need to optimize the concentration ratio of each excipient. For this process, we used design of experiment approach. We used optimal mixture design and formulated 13 formulations containing 13 different ratios of all the excipients. These formulations were characterized for size, PDA, and transmittance, and based upon these responses, a final optimized formula was derived at the end of DOE trial. Moving to the screening of precipitation inhibitor, the normal in vitro supersaturation test do not take into consideration the digestion process. But in lipid-based formulation, digestion plays a crucial role in determining the amount of drug which will be available for absorption. Hence, we used an alternative approach for screening of precipitation inhibitors, that is the high throughput in vitro lipolysis method. In this method, we prepared formulation containing different precipitation inhibitors and we subjected them to digestion process. At regular intervals, samples were withdrawn and the samples were centrifuged. On centrifugation, we were able to get 3 distinct phases of undigested lipid phase, aqueous phase, and a pellet phase. Among these 3, the aqueous phase consists of drug which is present in the colloidal forms, and this is very crucial because only this aqueous phase concentration will be available for absorption. So we screen various precipitation inhibitors based on their ability to maintain high drug concentration in the aqueous phase for a longer period of time. We also calculated the supersaturation ratio. Based on these results, PVP-K30 was selected as the precipitation inhibitor. We prepared formulations with increasing concentrations of PVP-K30 and we subjected them to the HTP method. Based on the results, 2.5% PVP-K30 was finalized as the optimized precipitation inhibitor concentration. The final optimized formulation was characterized. It was found to have a nanometric droplet size and spherical emulsion globules. Further, the precipitates obtained from the lipolysis experiment was also characterized using SEM and PXRT. These analyses revealed that there was amorphization in the drug which was achieved using the formulation. The formulation was also found to be robust to dilutions, and it was found to be stable in simulated gastric and intestinal fluids. Coming to the drug release studies, the conventional dissolution method or the dialysis bag method will not hold appropriate for lipid-based formulations. As I earlier mentioned, the process of digestion is very crucial in determining the in vivo fate of lipid-based formulations. This led to the emergence of several different digestion protocols and different techniques for evaluating the in vivo fate of lipid-based formulations. On this note, we used a simultaneous lipolysis permeation method to study the drug release pattern from our formulation. In this method, we dispersed our formulation in the lipolysis media, and the digestion was initiated by adding pancreatin enzyme. On initiation of digestion, triglycerides, monoglycerides, and fatty acids will be produced. These fatty acids will cause reduction in the pH of the media, and this pH drop will be sensed by the pH electrode, and an equivalent quantity of NaOH will be added to maintain the pH to 6.4. The pH 6.4 has two significances. One is that activity of enzyme will be appropriate only at pH 6.4, and the other one is that the volume of NaOH consumed during the experiment will help us to predict the extent of digestion of our formulation. So at different time intervals, two sets of samples were withdrawn. One set was directly subjected to aqueous phase concentration determination, and the other set was transferred to the donor compartment of transdiffusion cell, and the permeation across dialysis membrane throughout the 3 hours was studied. The in vitro lipolysis results showed that our formulation was able to maintain 13.5-fold higher concentration in the aqueous phase compared to the suspension. The permeation profiles also revealed higher permeation achieved using the formulation. Further, we also calculated the steady state flux and apparent permeability, and this also revealed that the formulation showed significantly higher permeability coefficient compared to the suspension. Further, in vivo pharmacokinetic studies were performed on SD rats by oral administration of 10 mg per kg dose. These studies revealed that formulation was able to achieve 2.7-fold and 1.8-fold higher AUC and Cmax compared to the suspension. Establishing IV-IVC for a lipid-based drug delivery system is a complicated challenge, and it is still being debated. Even though many experiments and reports have explored the potential of pH-stat lipolysis method, still it lacks in establishing IV-IVC. Hence, in the current study, we combined the pH-stat lipolysis to a permeation setup, and we explored its potential in establishing IV-IVC. For this purpose, we calculated the cumulative permeated amount in vitro, and we compared it with the in vivo AUC. On plotting the in vitro and in vivo AUC of all 3 formulations, we were able to get a linear correlation with R-square 0.9042. To conclude, the developed supersaturable biosnits was found to be a potential drug delivery strategy to improve the solubility and bioavailability of Dasatinib. And the explored simultaneous lipolysis permeation method was found to be a promising tool in estimating the in vivo performance of SNIDs. The future prospects of this project include application of mathematical models to establish better IV-IVC, performing in vitro cell culture studies using CAFO2 cell lines, evaluation of other model lipid-based formulations using this method in order to check the reproducibility of the method, and using the supersaturation strategy for other anti-cancer drugs as well. Thank you for your patient listening.